Welcome to Parasitic Diseases. Today's topic is Trichinellosis, an infection of humans and other animals caused by Trichinella spiralis and its relatives. When Trichinella was first discovered, it was thought that there was only one species, Trichinella spiralis, and for many years we labored under that pretense that indeed what we were looking at was a single species of worm which was distributed throughout the world. Well, as it turns out, with modern technologies and DNA to back us up, we can now talk about at least 8 different species of Trichinella, and many of them are restricted to various geographic regions. This map shows you the distribution of all of the known species of Trichinella, but the one that we'll be concerned with today is the one that causes most of the infections in humans, and that's Trichinella spiralis. Nonetheless, we still have to contend with Trichinella nativa, Trichinella britovi, Trichinella pseudospiralis. Then there are 2 that have yet to receive names, but we know that they're separate because of their DNA biology. It's totally different than the other 8 species that we know of. Then Trichinella nelsoni, which is confined to the continent of Africa. Trichinella papuae, which is found in Papua New Guinea. And of course, as you might be able to guess by now, Trichinella zimbabweensis is found in Zimbabwe. The discovery of this parasite is rather famous because it involves 2 well-known characters in medical history. Actually, one of them is not quite a medical history person, namely Richard Owen. Richard Owen was the director of the British Museum of Natural History back in the early 1800s, back in the age of the discovery of dinosaurs. Now, everybody knows about dinosaurs, but I bet you didn't know that the term dinosaur means terrible lizard. And I bet you also didn't know that that word was coined by none other than Richard Owen. Ironically, Richard Owen was not a believer, and in this case, it's not a belief system, but I'll just say he's not a believer in evolution. And he was the one that opposed Darwin's theory that life can change due to its adaptation to changing environments. Of course, the change is random, but the adaptations are not. And so what we can find is a series of animals and plants distributed throughout the world, all in tune, basically, with their environment. Some live in cold environments, others live in warm environments, etc., etc. Well, during the early formation of that theory, while Darwin was still active, Richard Owen was an opponent. But nonetheless, he was also a very active biologist and discovered many, many things, including Trichinella spiralis. Well, he didn't actually discover it. He co-discovered it. So now we have 2 people working both in England. One was a first-year medical student, James Paget, and the other was the vaulted director of the most prestigious Museum of Natural History that the world had known up to that point, the British Museum. And as the story goes, a piece of diaphragmatic tissue from a patient who had died of tuberculosis came to St. Bartholomew's Hospital. Paget was a first-year medical student, and he inquired as to why the scalpels of the surgeons kept dulling as they tried to cut into this cadaver. And as it turned out, it was because of a condition known as sandy diaphragm. That's the response he got from the surgeons, at least. And then, of course, he had to inquire as to what causes sandy diaphragm. And they almost vilified him in public for being such a curious person. How dare you ask us these questions while we're working? And while the surgeons went off to lunch, Paget snuck back into the autopsy room, grabbed a piece of sandy diaphragm, and ran it over to the British Museum of Natural History to examine it under high power using a microscope, which had recently been acquired by a gentleman who became famous for his Brownian motion, Dr. Brown. Well, at the same time, someone from that autopsy room obtained a similar piece of tissue and ran it over to the same museum. And instead of going to Brown, of course, he went to Owen. So two people in the same museum, unbeknownst to the other, looked under the microscope and co-identified Trichinella as the cause of sandy diaphragm. And you see the original drawings of Owen are shown here, and he almost drew them to scale so that you can almost not see the details of what he was seeing through his microscope. But indeed, he was given credit because he wrote up his results and published them in the Royal Society. Owen, however, was unaware of the fact that James Paget wrote letters home to his brother almost every day, telling him what he had learned in school that day. And that day he wrote back and said, a remarkable thing happened today. A patient who died of tuberculosis, or consumption as they called it, suffering from sandy diaphragm. I took a piece of his tissue over to the museum. I looked under a microscope, and lo and behold, what did I see inside? Little worms coiled up inside little dense bodies, some of which looked as though they had become calcified. So this dates the actual discovery to the same day, although Richard Owen was not prone to giving Paget much credit for his discovery because obviously he wasn't unaware of the fact that he had done that. So today we're going to discuss all of the life cycle and pathological ramifications of this worm. And indeed, we are indebted to the work of Rudolf Virchow, who worked tirelessly in his laboratory in Germany in the middle 1800s to describe the life cycle of Trichinella. It's got male and female worms. And remember, hookworm had the unusual bursa at the end of the male, which it used to grab onto the female with during mating. Trichinella has a similar apparatus. It's not exactly called a bursa. It's called claspers. And the worm mates with the female only briefly, inserting his spicules at the vulvar level of the vagina, which is shown right here. He inserts his sperm. They're captured by the female, which she uses then to fertilize the eggs, which start in the ovary here. And the worms can be shown in embryological development all throughout the uterus, and they're born live. And here's a scanning, or rather a phase contrast microscope picture of the newborn larva. This is the head end of that worm, and here's the tail. And she's filled with these worms, and indeed she can produce approximately 1,500 to 2,000 offspring before the immune system catches on to what's going on and expels the worms. These little worms stay within the same host, and lo and behold, 20 days later, transformed into the infective larva for the next host. So here we are dealing with a parasite which actually infects two different hosts, and yet the sex of the worms are found in both of these hosts. So they're both definitive hosts, and they can involve a variety of animals. Trichinella, spiralis, and its relatives infect almost every mammalian species on the planet, including what we thought were strict herbivores, but indeed, as we'll see, that's not always the case. The worm itself lives in a similar place that Strongyloides adults live in. The adult worm actually lives embedded in a row of columnar cells throughout the small intestine, and you see that depicted here. A cross-section of a villus shows the cross-section of the worm here, and the cell is stretched to its breaking point by the diameter of the worm, but nonetheless it's able to barely fit within this intramulticellular niche, and there it lives and gives birth to live larvae. So let's look at the life cycle now. It begins when an animal, including us, ingests a piece of infected muscle tissue. And this is not any ordinary muscle tissue. It's not cardiac muscle. It's not smooth muscle. It's striated skeletal muscle. Now, minus the bones, most mammals are about 40% in their weight of striated skeletal muscle. So it represents a very large portion of an organ system. The worms live coiled up inside of these individual units, which I will later on describe in detail. And there they sit. And how long can they sit for once they are coiled up and ready to go into the next host? And the answer is for many months to years. So this is a dormant stage of the infection, which in an animal that harbors them can last for almost the lifespan of most small mammals and medium-sized mammals. When you get up into the sizes, of course, of horses and elephants and other mammals, the bigger the animal, the longer the life. But many, many small mammals scavenge in the environment, ingesting larvae from another infected animal that might have died of natural causes. And as a result, they acquire their infections and pass this infection on from animal to animal, and that's the way it's maintained in nature. Now, for human environments, of course, this life cycle involves domesticated animals that we eat on a regular basis. And in fact, the animal of choice in this particular case is represented by the pig and its immediate relatives. So there are many varieties of pigs found throughout the world. Pigs are easy to grow because they are omnivores. They'll eat anything, and many pig populations are peridomestic. They live among people rather than next to them, and people feed little scraps of meat to their pigs to make sure that they grow up nice and big. And as the result of that behavior, the Trichinella spiralis infection has been maintained close to where people live. And of course, when they slaughter the pig and they eat it, well, this is what happens. So let's imagine, for instance, this is a piece of pork, and it's been freshly slaughtered or refrigerated without being frozen, and that's very important. We'll come back to that. The larva inside is all set to go. It's swallowed. The muscle tissue is digested away from the worm in the stomach, and the worm itself is then released and travels to the small intestine. When it gets to the small intestine, it actually burrows into the row of columnar cells that the adult ends up living in and undergoes molts. Now, it was unsure for many years as to whether this was a third stage worm, which is typical for nematodes as the infectious stage, or whether it's a second stage or perhaps even a first stage worm. So what we can't tell by just looking at this entity is to whether or not it's actually undergone a molt. But the biology strongly suggests that once this worm completes its life cycle and enters a muscle cell and develops this unit, it has not molted. So this represents the only example that I can think of in which a first stage larva becomes the infectious stage for the next host. So let's just assume for the moment that that's true. But if some evidence shows up between now and the next presentation, I'll be happy to correct. We've got a first stage larva here that now starts its growth cycle. And I myself, through my own research, have found, because this is the organism that I conducted a lot of personal research on as a researcher in the laboratory, I have evidence for the molts for this worm in the tissues as it begins its life cycle. And it's a rapid life cycle. This worm molts 4 times within 36 hours. That's a world record as far as I know. I don't know any other worm that molts that fast, moving from L1 stage to L4 to adult in 36 hours. Within 2 days, if one were to, in an experimental infection, let's say in mice, take out the worms and see which ones are male and which ones are female and whether all the females had mated or not, the answer would be within 2 days, all of the females have mated. What makes that unusual is the fact that when these worms are ingested, the ratio of females to males is 2 to 1. So there are twice as many female worms in a Trichinella infection, at least we think so in terms of human infection, because we obviously can't do those experiments, but in every other animal model that we've used to examine the biology of this infection, the ratio has always been 2 females to every male. So within 2 days, all of the males have mated with at least 2 worms on average. The females then become obviously pregnant with young, and because the young are born live over the next 2 to 3 weeks, they're born sequentially. So every larva in the body that's produced as the result of their exiting from the female worm and eventually getting involved in the bloodstream, which happens in the villus tissue, by the way, Every worm is of a unique age, so that no two worms are alike in a normal infection. I'll describe a situation later which can result from a synchronized infection. And at that point, almost all the larvae that end up in the muscle tissue are of the same age, and therefore we can look at the development that way. But that's jumping ahead a little bit. So when the female worms begin to shed larvae, the larvae go somewhere. Now, with Strongyloides, remember, when the females laid their eggs and the larvae developed, they then penetrated out into the lumen of the small intestine and passed out in the feces. Now, that doesn't happen with Trichinella. This live-bearing parasite sheds its worms. They get involved in the lamina propria. They then have an option of either penetrating the capillary that's there or the lymphatic vessel that's there. And most of them end up in the blood supply, but a good portion of them also go to the lymph. Eventually, of course, they all end up in the blood. They then circulate throughout the body, and when they reach a capillary, they get stuck and they penetrate out. Now, this sounds a little bit like the ascaris larva, which gets stuck in a capillary in the liver first and then later on in the lungs. In this case, it doesn't matter which organ this larva gets stuck in, it penetrates out and goes into a cell, which causes a lot of damage, by the way, given that there are 1,500 to 2,000 of these larvae being produced over a 3-week period per adult. What happens to the cell? The cell, well, it depends on which cell. The cell of not of choice, because this implies some directionality to the infection, but by luck, because muscle tissue represents 40% of the wet weight of an animal, minus the bones. The likelihood is that in 40% of the time, it's probably a little bit more than that, the worms penetrate a muscle cell, a striated skeletal muscle cell. And when that happens, the larva actually recognizes through a feedback loop, which obviously involves chemoreception and thigmotactic reception and perhaps some other sensory organelles as well, because the worm has a fully developed nervous system even in the larva. It stays put. It knows it's in the right place. And so this is called site selection for the niche that it's going to occupy for the next, perhaps, 3 to 5 years, depending on the age of the animal. The larva fully extends its body into the cell. And as you'll recall, a striated skeletal muscle cell is a long tube filled with nuclei that are spread evenly throughout the internal sarcolemma of the muscle tissue. So this worm occupies a very small portion of a single muscle fiber. And then some remarkable things happen, which I might supply details for in a moment. But what I really want to say is that this worm then begins its growth cycle. And the growth cycle involves the maturation of a row of cells in the worm called stichocyte cells. And these cells are secretory in nature, and they contain secretory granules of various kinds, much the same as a pancreatic cell. The acinar cells contain secretory granules that we're more familiar with, of course. And these granules each have functions because they're loaded with different proteins per granule type, and each one is released at a certain time during the maturation process of this first stage larva, early entry, 20 days later, to this mature unit, which is now fully encapsulated and has elicited its own blood supply. It's quite a remarkable structure, actually. And this worm is now fully ready to go into another animal. This process is called nurse cell development. The larva inside elicits all of these changes in host cell differentiation into this cell type that's never been observed before in that animal. The fact that this cell has not been observed inside the animal doesn't mean it's possible to make that cell, because we know that there are various kinds of cancers that result in cell morphology changes and cell function changes that relate very much to the agents that cause the cancer. This is not a cancer, but this is certainly a cell transformation process engineered by the parasite but all of the engineering takes place on the part of the host to respond to those signals that the parasite gives off over the 20-day period that it takes for this worm to mature from this stage to a mature infectious larva. Very complicated biology. The pathological consequences of this infection are all related to the fact that this worm has no idea of where it is until it enters a cell. So it's not directed towards the muscle cells by some chemoattractants or some activity-based signaling that might go on between the parasite and the host. It randomly seeks out a cell once it gets to a capillary, and if it's the wrong cell, it just penetrates right back into the capillary and continues its journey until it completes it. As a result, there's a lot of central nervous system damage done by worms migrating into brain tissue, damaging the cells, perhaps even killing them, and then migrating back out again. And the same is true for cardiac tissue. It will migrate into cardiac muscle, but it's the wrong muscle. As a result, no nerve cells are formed. The larva backs out again, seeking the right kind of cell. And indeed, this parasite in large numbers can induce failure in the host. And failure is due primarily to cardiac arrest or central nervous system failure. The resulting infectious unit, known as the nurse cell larva complex, is shown here, and it is quite an elegant structure. The worm is shown here, and that's why Owen and Paget thought of this as a spiral. And in fact, Owen named this Trichinella spiralis. It turns out that there was another parasite, or another organism rather, named Trichinella. So it's a little hair, which that means. So we altered the name after he named it to Trichinella to reflect that association. All of the other tissue that you see here is host. Quite a remarkable arrangement indeed. There are no parasite equivalents to this other than the Trichinella family of parasites. It's a complicated picture which, simplified, merely represents all of the damage that this larva causes in its ability to penetrate in and out of cells. And as this nurse cell develops, as I mentioned before, during the process of maturation, around day 12, because we can synchronize the infection by simply collecting these newborn larvae and injecting them into a muscle tissue, that synchronizes the infection. If we isolate on a daily basis tissues and look at what's going on, we see by day 12, we've noticed that the worm has attracted a circulatory ready. This circulatory ready supplies nutrients for the growing parasite and takes waste products away, just like it does for us. Except in this case, it functions solely for the benefit of the parasite. This observation was made back in 1865 by Pagenstecker, who worked for Virchow. Remember, Virchow did most of the life cycle work, and his assistant, Pagenstecker, actually said, you know, I wonder what would happen if I injected India ink into this animal. I wonder if I could reveal some of the structure of the worm this way, perhaps. And in fact, what he did was he lit up, in quotes, lit up the circulatory ready, which for many years was not understood as to what they were actually looking at. But in fact, using anatomical plastic casts, we've now shown that this is indeed a circulatory ready that the parasite somehow induces to form from host circulatory system. So the disease can be linked to the stage of the infection. Notice we start with death as the main criteria for the severity of this infection. And this gray zone, shown all the way down in this chart, shows the time at which, if the patient eats so many worms that they will eventually succumb to the infection, they will die between week 3 and week 6 of the infection. And all of these changes that you see listed on the side over here, creatine phosphokinase, which is a muscle protein that's released during the early migration of the worm into the muscle tissue, the production of muscle larvae shown here as they mature, whether it's immunologically responsible or not, the patient has responded. If you look before the second week, you will not find an antibody response that's detectable. And it becomes largely detectable after the third week and remains so throughout the life of the infection. So just knowing that it's positive doesn't tell you whether the patient has active infection or not, which is true for so many other infections as well. Notice that we've graded this graph in a light green, a darker green, and darkest green, and it shows an increasing height of response. This is a mild infection, a moderate infection, and a heavy infection. And we get production of eosinophilia, circulating eosinophils, which actually turn out to be essential for the growth of the parasite, an unexpected result that was recently discovered. So eosinophils are actually useful for the parasite, as I mentioned, a result which for many, many years was thought to be an immunologically based response that the host was using to fight off the parasite. And it turns out that if you ablate the eosinophils altogether, the parasite doesn't grow very well. We have symptoms and signs to look at. We have conjunctivitis and periorbital edema, fever, muscle pain, diarrhea. And notice the diarrhea comes very early on in the infection, during the period of time when the adult worms are present. When the adult worms are finally kicked out by the immune response, the diarrhea disappears. So the early diagnosis of this infection might very well be diarrheal disease caused by bacteria or protozoans or viral infections, and it actually turns out to be largely or solely due to the presence of Trichinella adults. We have late onset consequences of infection, mostly involving central nervous system disorder and heart function. The rest of this all deals with the various stages and when they're present in the host, and then you can match up which stages of the infection match best with which signs and symptoms and tests of various sorts. So making the diagnosis of this infection is not straightforward. You can't find eggs of this parasite in the stool. You can't find larvae in the stool. If you look in the stool, you'll look forever and you won't find anything. So you have to do other things in order to rule in this infection. The signs and symptoms include bilateral periorbital edema and the fact that the worm is penetrating out of capillaries all over the body. You can find splinter hemorrhages under the fingernails as shown here. And you can see that the sclera of the eye is all red. That's due to the leakage of blood into the spaces, into the intracellular spaces as the parasite penetrates out of a muscle cell or into a cell of various kinds throughout the body. Every time it does that, that represents a bleeding event. You could, of course, biopsy muscle tissue. If you did so during the early phases of development of the nurse cell, no pathologist could possibly recognize this little larva stuck in the middle of a muscle cell. But as the progression of nurse cell formation proceeds, it becomes more and more recognizable as a unit that finally, when it's mature, is shown here. Notice the intense inflammation around the outside of this worm. A lot of these cells are eosinophils. But other cell types are there too. Macrophages, in some cases neutrophils as well, and plasma cells. But mostly it's an eosinophilic response to the worm itself. And now Dr. Daniel Griffin is going to give a clinical vignette illustrating the pathogenesis of this infection.
A man in his 30s is admitted to the hospital up in Anchorage, Alaska in early June. His chief complaint is severe muscle pain and tenderness. He reports that he's usually very healthy, but about 1 week ago, he had a bad case of diarrhea with belly pain and vomiting. This lasted for a full week, and then he felt better. Now he is having fever, and his muscles just all hurt. He is a hunter and did just come back from a successful black bear hunt. On exam, he appears anxious, he has swelling around his eyes, and his proximal muscles are tender. Laboratory investigations reveal white blood cell count 14,000 with 30% eosinophils. His basic metabolic panel is unremarkable, but his CK, creatinine kinase, so his muscle enzymes are elevated, his LDH is elevated. Let's talk a little bit about clinical disease. While light infection with Trichinella can pass unnoticed with minimal or no identifiable symptoms, heavy infection usually presents as two distinct stages, so a two-stage syndrome, a gastrointestinal stage and a parenteral phase. So let's talk first about the gastrointestinal stage. The first few days of infection are characterized by this gastrointestinal stage with gastroenteritis, secretory diarrhea, abdominal pain, vomiting. Now, this phase is transitory, usually lasts less than 10 days after ingestion of the infected tissue. Now, a history of eating raw or undercooked meats, as Dr. Despommier has introduced you to, is something that raises concern for this parasitic infection. Also, if others who ate the same meats are suffering similar symptoms, that also reinforces the suspicion. Now, the second, the parenteral phase, this begins about a week after infection and may last for several weeks. The patient might have fever, might have muscle pains, might have diffuse and particularly periorbital edema, petechial hemorrhages, which are often most clearly seen under the nails, the subungual region. But you also can see these in the subconjunctiva and other mucosal sites. The muscle tenderness can be readily detected. Laboratory studies often reveal an elevated white count and a rather significant elevation of the eosinophils, sometimes to as high as 50%. Larvae penetrating the tissues other than skeletal muscle can give rise to serious sequelae. Now, in many cases, you can have myocardial involvement, so cardiovascular involvement. It's been suggested, though, that this is overrated. Most presentations are going to be of a milder variety, so this will not be seen probably as frequently as was suggested, but it is something to be concerned about. Now, in up to a quarter of patients with heavy infection, you can actually get meningitis, encephalitis. You can end up with infarcts and even hemorrhage in cases that might go on to be fatal. After these two stages, there's a convalescent phase. This follows the acute phase, and it's actually during this time when many, but not all of the nurse cell parasite complexes are destroyed. And in general, this whole presentation, the severity, is related to the amount of ingested larvae, the species of Trichinella, and the status of the infected host. This is a nice time course of how we see it. So the incubation, the enteral phase, and then it's going to move on into the parenteral phase. And you can see where the diarrhea symptoms, where the muscle pain symptoms, just sort of a nice chart for the progression over time, over weeks after ingestion of when people develop all the symptoms. You can see the eosinophilia has a really broad hump there. Now, a couple of the clinical signs, the splinter hemorrhages that we talked about here, and the periorbital edema. There's a limited number of things that will cause that, so this is one of the suggestions that we might see clinically. And here is an image of the larvae penetrating a muscle fiber. Now, what about diagnosis? Microscopic identification. Definitive diagnosis is if you're actually able to find the nurse cell parasite complex in muscle biopsy, or you're able to get a specific DNA amplification by PCR. Now, PCR is sensitive and specific, but it's not as available in the clinical setting as one might hope. Muscle biopsy is a challenge because it can be negative even in the heaviest infection because of the sampling area. You really want to focus on a proximal symptomatic muscle, such as the deltoid, particularly near the tendinous insertion. This tends to give us the highest sensitivity. Also, there's a timing issue because depending upon when you do it, the larvae may be at an early stage of development, and this makes it a little harder to recognize even for an experienced pathologist. There are some serological tests, and these can be confirmed with Western blot, but they're going to take about 2 weeks to turn positive. So you want to wait about 12, 14 days after infection is when you start to be able to detect these antibodies. There's a number of supportive laboratory results, so the muscle enzymes, the peripheral eosinophilia, But then it's helpful to remember that it's not the pork, which I think we grew up with, but actually a lot of trichinosis has been traced to hunters, and then people that are sharing in the ingestion of those kills. Here's a muscle biopsy of an old infection. Now, what about treatment? Now, since the larvae may still be burrowing through the gastrointestinal tract when patients present with symptoms, it's actually recommended that people go ahead and treat with albendazole or mebendazole. And I say may be beneficial because we are not sure, but that's the idea. Now, treatment with mebendazole early in the course has been shown in some studies to be associated with decreased muscle pain and muscle inflammation. So, in general, it's what we recommend. Now, albendazole would be 400 milligrams twice a day for a couple weeks. Mebendazole would be 400 milligrams 3 times a day for a couple weeks. And in some cases, people will add prednisone as well. If you remember back to some of the discussion that Dr. Despommier brought in the molecular and cellular aspects of this, there are some interesting issues about how that may affect the ability of the larvae to go ahead and form the nerve cell complexes. Now, once muscle invasion has occurred, treatment with these agents will not succeed in destroying the encysted larvae. So if someone is suffering from significant amounts of inflammation, then anti-inflammatory medications such as steroids, prednisolone, if the diagnosis is secure, we're not worried about something else, these can be considered. Now, the myopathic phase, in addition to steroids, antipyretics, analgesics, so aspirin, acetaminophen, these are often continued until the fever allergic signs recede. But as mentioned, because the immunosuppressive potential, steroids, when considered, should be used with caution when one is confident of the diagnosis. Now, what about our patient? Now, our patient had an ELISA that was positive for Trichinella antibodies, underwent a muscle biopsy of the left deltoid right near the tendinous insertion, as we would suggest, and they were able to detect early stage larvae. PCR was not done, and the patient recovered. There was no specific treatment.
So the best way to avoid Trichinella infection to begin with is by prevention and controlling the source of infection so that no commercially raised meat products reach the consumer. We have individually, you could cook thoroughly or freeze all meat products, and in that way you would avoid infection with a variety of food-transmitted diseases, including Trichinella, of course. Not everybody likes their meat well done. And there's certainly a movement afoot in most of the developed world for fresh produce, not frozen. And this avoids all of the recommendations of an attempt to avoid contact with this infection to begin with. So these, while they're great advices to people while they're contemplating what they'll have for dinner that night, you can't generally apply this to modern society. That's really too bad, but it really doesn't work out that way at all. You could, of course, print out a warning on all big game hunting licenses, which is being done in North America at least, that all animals that are killed and are consumed should be cooked well, and not rare or medium rare. There is a caveat here too. Freezing the meat of a wild animal does not kill the larvae in their muscle tissue, as it does in pork, which is the domestic pig. So even if you knew to cook your game animals, it's probable that you'd chop it up into big pieces first and you'd freeze it, and you say, oh, it's okay because I froze it. I know what to do for the pork products, and I'll just freeze it first. That doesn't work. Apparently there's an antifreeze molecule associated with wild animals that actually protects ice crystals from forming inside of the nerve cells, and it protects the worm as well as it does the animal. There is another approach to this, of course, and that is on a community basis. You can have meat inspection. You can actually look in the diaphragm tissue, as Owen and Paget did, and you can identify these worms as they're coiled up inside their nerve cells. A large circle of diaphragm tissue is cut out from each pig that's slaughtered, and placed between two very large clear lucite plates, and it's screwed tight until the tissue inside is flattened and expands out. And then experts could actually just hold this up to the light and see these little white flecks throughout the meat. Upon microscopic examination, they're shown to be Trichinella. There's a special device that this whole apparatus slides into, and it's projected up on a screen. It's called a Trichinelloscope. And you get this diaphragm tissue that's very greatly enlarged. And inside of the tissue, if it's positive, you can even spot the 1 or 2 nerve cells that happen to be there. Heavier infections, of course, show up as multiple nerve cells. There's a lot of recommendations for preventing the spread of this infection from animal to animal on a domestic level. Pigs, for instance, are no longer fed meat scraps. At least in the United States, it's against the law. But it wasn't instituted to prevent the spread of Trichinella. It was instead hog cholera that they were trying to prevent, a viral infection that's extremely infectious and damages entire herds of pigs and renders them unedible. And as a result, hog cholera has caused great economic loss in the United States. This law was instituted to prevent the spread of hog cholera by, first of all, cooking the meat scraps, and then you can feed them to the pigs. But before you do that, you're running a big risk of spreading cholera. If you're caught as a producer, the fines are incredibly heavy. They're so heavy that they could even put you out of business, because hog cholera spreads very rapidly from pig population to other pig populations. But inadvertently, I would say, in instituting this law, we've also gotten rid of Trichinella because it's killed in exactly the same way. So within the United States, in the year 2016, there were very few cases of Trichinella recorded within the United States, and none of them were due to the ingestion of commercially available pork products produced by commercial pig farms. So we've essentially eliminated the infection from domestic pigs in this country, but remember, the game animals out there are still circulating those wild forms, not necessarily Trichinella spiralis, but certainly the ones that are capable of infecting people as well. So we always have to be on the vigilance for this. One interesting note is that with the fall of the Russian Empire, so to speak, when the Iron Curtain finally did come down and a lot of Central European countries regained their independence, they reverted back to an older form of farming because the Russians had realized that this life cycle is ongoing, and a lot of those pork products made its way from Central Europe back to Russia. And Russia didn't want to have to contend with Trichinella, so they were very strict about enforcing these rules. Once the Iron Curtain was lowered, the traditional farmers returned to their old ways, and there was a resurgence of trichинellosis in people within Central Europe as the result of the relaxation of these rules. So we know they work when they're enforced. There's a nice review paper recently written by a friend of mine, Darwin Murrell, and he's such a famous person that worked for the United States Department of Agriculture, and he and I collaborated on several papers together, but mostly he's devoted his entire life to studying Trichinella, that there is now a species of Trichinella named after him, Trichinella murrelli, which can infect people and cause disease as well. So here's a nice review article written by him, and we've had, at least 4 episodes of TWIP in which Trichinella has been a featured player in our discussion, and you can access that, of course, on microbe.tv/TWIP. Next time, we're going to discuss lymphatic filariasis caused by Wuchereria bancrofti and Brugia malayi. Thanks for listening.